Magnetic field effects on plant growth, development, and evolution. The geomagnetic field is a natural component of our environment. Plants, which are known to sense different wavelengths of light, respond to gravity, react to touch and electrical signaling, cannot escape the effect of GMF, geomagnetic field, while phototropism, gravitropism, and stigmatropism have been thoroughly studied. The impact of GMF, geomagnetic field, on plant growth and development is not well understood. This review describes the effects of altering magnetic field conditions on plants by considering plant responses to MF values either lower or higher than those of the GMF, the possible role of GMF on plant evolution, and the nature of magnetoreceptor is also discussed. The point of this study is basically showing that magnetic fields affect plant growth, sometimes positive, sometimes negative. I'm going to read another abstract, a similar idea, coming to the same conclusion. Magnetic fields affect plant growth. The title of that study is Magnetic Field Regulates Plant Functions, Growth, and Enhances Tolerance Against Environmental Stresses. The abstract, which is a quick summary, is this. Global climatic fluctuations and the increasing populations have been responsible for the decline in the crop productivity. The chemical fertilizers, pesticides, and suitable genetic resources are commonly used for improving the crop yields. Magnetic field (MF) therapy for plants and animals have been found to be an effective and emerging tool to control diseases and increase tolerance against the adverse environment. Very limited studies have been attempted to determine the role of MF on plant tolerance against various stress conditions. This review aims to highlight the mitigating effect of MF on plants against abiotic and biotic stresses. MF interacts with seeds and plants and accelerates metabolism, which leads to an improved germination. The primary and secondary metabolites, enzyme activities, uptake of nutrient and water are reprogrammed to stimulate the plant growth. Sorry about that, I just got a weird call. To stimulate the plant growth and yield under favorable conditions. During adverse conditions of abiotic stress, such as drought, salt, heavy metal contamination in soil, MF mitigates the stress effects by increasing antioxidants and reducing oxidative stress in plants. The stunted plant growth under different light and temperature conditions can be overcome by the exposure to MF. An MF treatment lowers the disease index of plants due to the modulation of calcium signaling and proline and polyamines pathways. This review explores the basic and recent information about the impact of MF on plant survival against the adverse environment and emphasizes that through research is required to elucidate the mechanism of its interaction to protect the plants from biotic and abiotic stress. And just a disclaimer, throughout this reading, when I see MF, I say magnetic frequency. Really, I should have said magnetic field. So just keep that in mind. Whenever I say magnetic frequency for MF, it's really magnetic field. I'm too lazy to edit all of it. I already recorded it all, so it's all right. Just keep that in mind. MF is magnetic field, not frequency, like how I'm saying. So I'm actually going to read the second study first because I think it's easier to understand. It's more simple than the first abstract that I just read. So just a heads up, the first half of this long video is going to be the second study I just read right now, and the second half of this long video is going to be the first abstract, which is more complicated. So if you want the complicated stuff, second half of this video. If you want the simple stuff, let's go ahead and dive into that right now. Introduction. The Earth is a giant magnet, and its geomagnetic field has a huge impact on the productivity of crops. Specifically, the electromagnetic radiations coming from the sun stimulate the growth and development of plants through the process of photosynthesis. The other possibility to increase plant growth could be an alteration an electrostatic balance of the plant system as the cell membrane level, as it is the primary of plant growth. The GMF can influence basic biological functions such as rhythmicity, orientation, and development. The effects of the magnetic field on plants, fungi, and microbes can be elucidated by ion cyclotron resonance, ICR, and the radical pair model. These two mechanisms also play an essential role in the magnetoreception of organisms. Many scientists have proposed various theories about the biological effects of MF. These are as follows. A moving electric charge generates an MF around it, like an electron, ion, or polarized particle. The organic material constituting living organisms has a polar structure due to various polarized chemical bonds, which may be linked to water molecules and dissociated mineral salts, conferring magnetic properties. The hypothetical interaction of weak magnetic fields, WMF, weak magnetic fields, and the living organisms has been proposed by Binney. The experimental analysis of electromagnetic fields, EMF, applied to biological systems has gained a rapid interest in the past few years. The application of MF are being explored in several areas, particularly in the agricultural science. The effect of MF on seed germination, biochemical, hormonal changes, plant growth, and yield have been subject to several investigations. The enhancement of growth in crops under precise magnetic conditions has been confirmed, but a systematic and extensive study is still necessary to delineate the mechanisms of magnetic action in cells and tissues. Although attempts have been made to understand the mechanisms of action of extremely low frequency EMFs in biological systems, still more detailed studies need to be undertaken. An application of 20 to 30 MT of MF on crop plants revealed an enhancement in their growth. The plant cells contain ferritin, and each cell has about 4,500 iron atoms involved in growth and metabolism. The magnetic rotator moment of ultimate iron atoms creates an external MF and collectively generates oscillations, which generate energy and finally reposition the atoms in the direction of MF. This increases the temperature in plants, which depends upon the duration and the frequency of MF treatment. WMF modulates cryptochrome and phytochrome mediated plant responses in plants. Very limited information is available on the molecular basis and the function of the putative WMF receptors and their activation by physiological signals. Therefore, their involvement in directing the overall response in different plant organs is yet to be determined. Sevostin first reported a twofold increase in wheat seedling elongation under MF. Murphy observed the positive effects of MF on seed germination. Adis and Pittman also studied a strong magnetropic effect on root development. MF influences the normal tendency of Fe and Co atoms and utilizes their energies to continue the translocation of microelements in root meristems, which leads to an increased plant growth. The different dosage of MF alters the root biomass, stems girth, and leaf size. Further, the root growth is more sensitive than shoots to MF. The pre-treatment of seeds by MF resulted in seedling growth, seed vigor, and increased crop yield. MF accelerates growth by triggering the protein synthesis and activates the root tropism by altering the intercellular movement of amyloplasts in the statocysts of root cap cells. A positive effect on seed germination, uptake of nutrients, flowering, and crop yield can be achieved by applying MF. MF treatments also affect the plant metabolisms that involve free radicals and stimulate the activity of proteins and enzymes to enhance seed vigor. The effects of continuous as well as pulsed MF on plant growth and development have been investigated in a large number of plant species. Aladegian revealed that the exposure of MF 150MT stimulated shoot development, which led to an increase in the germination, fresh weight, and shoot length in maize. Is that corn? The mechanism of action of MF on plant growth promotion is still not very clearly understood. Therefore, an optimal external EMF may accelerate the plant growth, especially seed germination. Yinan et al. Observed a positive effect of MF pre-treatment on cucumber seedlings by stimulating seedling growth and development. The promotion of seed germination and the growth of plants depend on the magnetic flux densities, frequencies, and pre-treatment of the plant material. Several harsh environmental conditions such as drought, salinity, low or high temperatures, flood, pollution, radiation, and diseases are the important stress factors that adversely affect the growth, metabolism, and the yield of plants, and thereby limit the productivity of crops. The productivity of plants can be increased by the application of plant growth promoting substances, microbial inoculation to the soil, organic and inorganic manure, and several other non conventional approaches, such as plant breeding and genetic engineering. The application of MF is a novel approach to improve plant growth and the overall productivity. Plant growth promoting effect of magnetic field on plant physiology under favorable condition, seed germination. Many researchers reported an increase in seed germination under magnetic field exposure. Magnetic field stimulates the initial growth stages and early sprouting of seeds. Recently, Rad and Ron observed an increased rate of seed germination in soybean under pulsed magnetic fields. Morar et al. Also reported that MF influences the free radical formation and stimulates the activity of proteins and enzymes to enhance the seed vigor. The paramagnetic properties of plastid may be responsible for the enhanced seed vigor. MF increases the energy in plants and disperses this energy to biomolecules, which in turn stimulates the metabolism to enhance the seed germination. A metabolically active plant cell contains free radicals that play a vital role in electron transfer and the kinetics of biochemical reactions. These free radicals possesses non-paired electrons with magnetic activities that can be oriented under an external MF. The microwave energy is absorbed as a result of the interaction between the external magnetic field and the magnetic action of unpaired electrons. Finally, this energy is converted into a chemical form and accelerates the fundamental processes in seeds. The overall effects of magnetic field on crops are summarized in this figure here. Effect of magnetic field on function, changes in plants for their growth improvement. Magnetic field MF plus treatment triggers seed germination, plant growth, water and nutrient uptake, pigment synthesis, protein and enzyme activities as compared to the control. Vegetative growth phase. Magnetic field positively influences the growth of plants by increasing shoot and root length. Root growth depends upon the cell division in the root meristems and subsequent differentiation and elongation of the descendant cells. The root cap cells were notably larger and the metaxylem cells became significantly longer, starting from the quiescence center to periphery in MF treated plants. The induction of metaxylem cells by EMF is an important component of the increase in the rate of root elongation. Magnetic field exposure to seeds accelerates their growth, activates protein formation, and the root growth. In an experiment, sunflower seedlings exposed to magnetic fields showed a substantial increase in the shoot and root fresh weight, MF-treated plants also showed at the vegetative state a significantly larger leaf area and higher leaf dry weight than the controls. This effect may be attributed to the increased photosynthetic rates due to the better perception of light and nutrients available for vegetative growth. Reproductive growth phase. Very limited studies have documented the effect of MF on reproductive development in crops. Matsuda reported that magnetic frequency enhances the yield in strawberry. Similar effects were also witnessed for flax, buckwheat, pea, wheat, tomato, pepper, soybean, and cotton by Goebbels. Gabrielian, Firk, and Umbarker, Piet, Og, Vass, Lille, and Esset and Turan, respectively, and it was suggested that the enhancement in growth and yield in tomato plants may be attributed to an MF-induced energetic excitement of cellular proteins and carbohydrates and or water inside the dry seeds. Mitigation effect of MF on crops against unfavorable environments. The adverse environmental conditions, including drought, salinity, and heavy metal accumulation in soil and light temperature, insects, and pathogens, affect the growth and yield of agricultural crops. Magnetic frequency induced changes in the metabolism of plants during those unfavorable environments are given in the figure below. Biotic stresses. The application of MF can reduce the detrimental effect of pathogenic microbes and increase the growth of yield of plants. For example, citrus plants intermittently exposed to 10 Hz MF showed a substantial enhancement in fresh and dry leaf weight in healthy as well as Phytoplasma aurantifolia infected plants. It proved that MF could also increase the resistance against pathogens. Biochemical analysis revealed that the accumulation of proteins was higher, but carbohydrates were lesser in infected plants treated with MF. The synthesis of proline, a protective osmolyte, is notably activated by MF, thus supporting cellular structures. The biotic stress alleviation mechanism of MF was determined by reduced H2O2 production in infected plants exposed to magnetic frequencies. On the other hand, scavenging enzymes control the free radicals, which alter membrane integrity and increase the resistance in plants against pathogen infection. However, Treby et al. Studied the hypersensitive response HR in tobacco mosaic virus-infected tobacco plants during the MF exposure and proved that MF treatment decreases the number and area of lesions in the diseased plants, and it also regulates the calcium signaling pathway in the cell. During the HR, the CA2 influx into the cytosol is stimulated. That increases the resistance. That's strange. A strange way of wording it. Similarly, MF influences the polyamine pathway enzymes such as ornithine decarboxylase. I definitely didn't pronounce that right. And phenylalanine ammonaliase. The ODC and PAL activities enhanced by an MF exposure during infection suggests that putrescine synthesis helps the plant withstand the biotic stress. Conclusion. Magnetic field therapy has been claimed to be effective for human ailments. Very few studies have been undertaken to understand the positive effect of magnetic frequency on crop plants during environmental stress conditions. This review explores the current knowledge and future prospects of magnetic frequency-induced physiological changes in plants toward enhancing the growth and yield under favorable and adverse conditions. The application of magnetic frequency accelerates the seed germination, vegetative as well as reproductive growth in plants due to an increase in energy and its distribution to biomolecules in the cell.
The enhancement of water and nutrient uptake, photosynthesis, carbohydrates, protein, and enzyme metabolisms would impact the promotion of plant growth and yield. Unfavorable environments such as drought, salinity, heavy metal contamination in soil, cold and or hot conditions drastically decrease the crop productivity. MF exposed plants tolerate these adverse environments by reducing oxidative stresses. Magnetic frequency treatment can enhance plants' drought tolerance by stimulating water and Ca2 uptake. Cell membrane permeability, cambial differentiation, pigment synthesis, stomatal conductance. Similarly, MF protects the plant against salinity by increasing water uptake, stomatal conductance, sugar and protein synthesis, and also by regulating the antioxidants and defense metabolites. Heavy metals in soil suppress the plant growth, but MF treatment alleviates these metal stresses through the increased water flow. Nitrogen, carbon, endogenous, no accumulation, photosynthesis, stomatal conductance, transpiration, and cell division. In addition, the production of heat shock proteins in MF exposed plants confers protection against the hyperthermic stresses. During low temperature, MF triggers ion transport, membrane permeability, photosynthesis, stomatal conductance, and transpiration, and regulates the polar lipids and erucic where am I? Erucic acids, irrespective of the presence of absence of light conditions to enhance the plant tolerance against temperature stresses. However, a reduced area of infection in leaves showed the control of plant diseases by MF exposure, and this resistance may be due to the accumulation of Ca2 proteins and proline in plants. Future prospects. The MF induced changes in the fundamental physiological process of plants against adverse environmental conditions have been investigated by only few researchers. A comprehensive stimulatory activity of MF and several cellular metabolisms and their subsequent effects on tissue proliferation and organization need to be elucidated to decipher the mitigation mechanism of MF and plant interaction under stress environments. The future studies are required to confirm the positive effects of MF on crop yield by answering the following: whether magnetic frequency treatment influences the next generation of crop growth and yield. That's a good question. Is there any toxicity due to the consumption of magnetic frequency treated fluid? Another great question. Does it affect the micro and macro flora of soil during plant growth? In addition, the comprehensive genomic and proteomic analysis in magnetic frequency treated plants would also bridge the space between current understanding and future prospective of biological effects of the magnetic field in plants. Okay, now that we read that study, let's go back to that first study I read. It's a bit more complicated, but if you stuck around this long and if you want to learn more about this, let's dive into this study here. Intro. A magnetic field is an inescapable environmental factor for plants on Earth. During the evolution process, all living organisms experienced the action of the Earth's MF, geomagnetic GMF, which is a natural component of their environment. GMF is steadily acting on living systems and is known to influence many biological processes. There are significant local differences in the strength and direction of the Earth's magnetic field. At the surface of the Earth, the vertical component is maximal at the magnetic pole, amounting to about 67 UT, and is 0 at the magnetic equator. The horizontal component is maximal at the magnetic equator, about 33 UT. Sorry if I'm not saying UT properly. And is 0 at the magnetic poles. A MF is usually measured in terms of its magnetic induction, B, whose unit is given in Tesla. Sorry, that's my chair squeaking. The dimension of the magnetic flux, blah, 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 blah. The MF strength at the Earth's surface ranges from less than 30 UT in an area including most of South America and South Africa, to almost 70 UT around the magnetic poles in northern Canada and south of Australia.
and in part of Siberia. Most of the MF observed at the Earth's surface has an internal origin. It is mainly produced by the dynamo action of turbulent flows in the fluid metallic outer core of the planet, while little is due to external MFs located in the ionosphere and the magnetosphere. It is the presence of the GMF that, through the magnetosphere, protects the Earth, together with its biosphere, from the solar wind deflecting most of its charged particles. The literature describing the effects of weak MFs on living systems contains a plethora of contradictory reports, few successful independent replication studies, and a dearth of plausible biophysical interaction mechanisms. Most such investigations have been unsystematic, devoid of testable theoretical predictions, and ultimately unconvincing. The progress and status of research on the effect of MF on plant growth have been reviewed in the past years. Krylov and Tarakanova, 1960, were among the first to report on MF effects on plant. They proposed an auxin-like effect of the MF on germinating seeds by calling the effect magnetrotropism. The auxin-like effect of MF was also suggested to explain ripening of tomato fruits. There are evidence that the root growth response was not directly heliotropic but rather magnetropic or geomagnetropic. Observation of the roots of a number of other plants suggested that some inherent factors within a species, or even within a variety of a species, could also be necessary before the tropism becomes manifest. Because of the insufficient understanding of the biological action of MFs and its mechanism, it is rare to document the magnetic environment as a controlled factor for scientific experiment. Anyway, from these pioneering studies on, we can consider two experimental approaches aimed to evaluate the physiological responses of plant growth of plant exposed to either weak or strong MFs. Exposure of plants to a MF intensity lower than the geomagnetic field. The term weak or low MF is generally referred to the intensities from 100 nt to 0.5 mt, whereas super weak or conditionally zero, the so-called magnetic vacuum, is related to MFs below 100 nt. Investigations of low MF effects on biological systems have attracted attention of biologists for several reasons. For instance, interplanetary navigation will introduce man, animals, and plants in magnetic environments where the MF is near 1 nt. It is known that a galactic MF induction does not exceed 0.1 nt in the vicinity of the Sun, 0.21 nt, and on the Venus surface, 3 nt. This brought a new wave of interest in MF role in regulating plant growth and development. In laboratory, low MF have been created by different methods, including shielding surrounding the experimental zone by ferromagnetic metal plates with high magnetic permeability, which deviate MF and concentrate in the metal, and compensating by using Helmholtz coils. In general, developmental studies on plant responses have been performed at various MF intensities. Early in 1963, it was found that a MF of relatively low intensity could be effective in stimulating or initiating plant growth responses. Since then, a few experiments evaluated the effects of reduced MF conditions and compared to those performed by using high intense MFs, basically saying it seems lower magnetic field frequencies are better than higher, more intense magnetic field frequencies for plant growth. Effects on plant development. Sunflower seedlings exposed to 20 UT vertical MF showed small but significant increases in total fresh weights, shoot fresh weights, and root fresh weights, whereas dry weights and germination rates remained unaffected. Pea epicotals were lower in MF. Peas were longer in low MF, when compared to normal geomagnetic conditions. Elongated of P was confirmed by microscopic observations of section specimen to result from the elongation of cells, and osmotic pressure of seedlings was significantly higher in low MF than controls. This observation suggests that the promotion of cell elongation under low MF may relate to an increase of osmotic pressure in the cells. Furthermore, P seedlings showed ultrastructural peculiarities such as a noticeable accumulation of lipid bodies, development of a lytic compartment, and reduction of phytoferritin in plastids. Mitochondria were the most sensitive organelle to low MF treatment, and their size and relative volume in cells increased. I'm sorry, I got lost my place here. I was looking at my computer. Increased. Matrix was elongate, electron transparent, and Christie reduced. It was also observed that low MF effects on ultrastructure of root cells were due to disruptions in different metabolic systems, including effects on CA2 homeostasis. In broad bean seedlings, low MF intensities of 10 to 100 UT at 50 to 60 Hz were observed to alter membrane transport processes in root tips, whereas seeds of soybean exposed to pulsed MF of 1500 NT at 0.11 and 10 and 100 Hz for 5 hours per day for 20 days, induced by enclosure coil systems, significantly increased the rate of seed germination, while 10 and 100 Hz pulsed MFs showed the most effective response. Treatment with MF also improved germination-related parameters like water uptake, seed of germination, seedling length, fresh weight, dry weight, and vigor indices of soybean seeds under lab conditions. Geez, I gotta get better at reading. I suck. Controversial data also have been reported. The exposure to near null MF of different in vitro cultures of various species of the genus Solanum was either stimulating or inhibiting the growth of in vitro plants. The effect was apparently also dependent on the species genotype, type of initial explant, treatment duration, or even culture medium. By using ferromagnetic shields, the influence of weak alternating MF, which was adjusted to the cyclotron frequency of Ca2 plus K plus ions, if you care about this stuff, was studied on the fusion of tobacco and soybean protoplasts. It was observed that in these conditions, protoplast fusion increased in its frequency 2 to 3 times with the participation of calcium ions in the induction of protoplast fusion. The observations of the increase in the Ca2 CYT level after exposure to very low MF suggests that Ca2 entry into the cytosol might constitute an early MF sensing mechanism. Interesting. An early MF sensing mechanism. When wheat seeds were treated with low frequency, oh my goodness, what's wrong with me today? Frequency magnetic MF at the stage of esterase activation during seed swelling, the activation of esterases was, pronouncing a lot of this wrong, was enhanced by changing qualitatively the time course of the release of reaction products into the medium. These results helped to explain unusual dependencies of biological effect on the amplitude of the electromagnetic field, including the atypical enhancement of these effects by the action of weak, low-frequency fields. A 2-layer permalloy magnetic screen was used to test the effects of wide range of low MF, 20 nt to 0.1 mt, on 3 to 5 day old wheat seedlings. It was observed the seedlings grew slower than controls. Barley seedlings grown in Helmholtz coils with a 10 nt MF intensity showed a decrease in fresh weight of shoots by 12% and roots by 35%, as well as dry weight of shoots by 19% and roots by 48% in comparison with GMF controls. From the Pioneer study, it was concluded that very low MF was capable of delaying both organ formation and development. The effect of a combined MF at the resonance frequency of Ca2 ions inside a U metal shield and the altered gravitropic reaction of crest roots was performed to evaluate the structure and functional organization of root cap statocytes. The experimented conditions were observed to change normally positive gravitropic crest root to exhibit negative gravitropism. Artificial shielding of GMF caused a significant decrease in the cell number with enhanced DNA content in root and shoot of onion. Furthermore, the uncytokinetic mitosis with formation of binuclear and then tetranuclear cells, as well as a fusion of normal nuclei resulting in appearance of giant cells with vast nuclei, seems to dominate in very low MF conditions. Changes in the ultrastructural organization of some organelles and and cellular compartments, alterations in condensed chromatic distribution and reduction in volume of granular nucleosis components with the appearance of a nucleosis were also found. Okay. Effects on transition to flowering. Near null MF can be produced by 3 mutually perpendicular couples of Helmholtz coils and 3 sources of high precision direct current power, which can counteract the vertical north-south and east-west direction components of the geomagnetic field. Although the functions of cryptochrome have been well demonstrated for the effect of the GMF on the growth of Arabidopsis and its mechanism of action are poorly understood. Let's skip to the end. Therefore, the removal of the local GMF negatively affects the reproductive growth of them, which thus affects the yield and harvest. So basically saying the removal of the local GMF negatively affects them. Wish I read all of that to really understand it, but man, these words are just above my pay grade. I gotta really look up the definitions to a lot of these things. Exposure of plants to MF intensities higher than the geomagnetic field. A consistent number of papers described the effect of MF intensified intensities higher than the GMF levels. In general, intensities higher than GMF relate to values higher than 100 UT, as summarized in Table 1. Experimental values can reach very high MF levels, ranging from 500 UT up to 15 T. Most of the attention has been focused on seed germination of important crops like wheat, rice, and legumes. However, many other physiological effects on plants of high MF described plant responses in terms of growth, development, photosynthesis, and redox state. Effects on germination. A MF applied to dormant seeds was found to increase the rate of subsequent seedling growth of barley, corn, beans, wheat, certain tree fruits, and other tree species. Moreover, a low-frequency MF, 16 Hz, can be used as a method of post-harvest seed improvement for different plant species, especially for seeds of temperature-sensitive species germinating at low temperatures. Seed of hornwort exposed to sinusoidally time varying extremely low frequency MFs, AC fields, in combination with the local GMF showed a promoted activity of cells and enzymes in germination stage of the seed. Thus, this suggests that an optimum ELF MF, so ELF MF, might exist for the germination of hornwort seeds under the local GMF. In seeds of mung bean exposed in batches to static MFs of 87 to 226 intensity for 100 minutes, a linear increase in germination magnetic constant with increasing intensity of MF was found. Calculated values of mean germination time, mean germination rate coefficient, germination magnetic constant, transition time, water uptake, indicate that the impact of applied static MF improves the germination of mung beans even in off season. So basically let me skip around here. Seeds of wheat were imbibed in water overnight and then treated with or without 30 MT static magnetic field SMF and a 10 kilohertz EMF for 4 days, each 5 hours. Exposure to both MF increased the speed of germination compared to the control group, suggesting promotional effects of EMFs on membrane integrity and growth characteristics of wheat seedlings. Effects on roots and shoots. Increased growth rates have been observed in different species when seeds were treated with increased MF. Treated corn plants grew higher and heavier than control, corresponding with increase of the total fresh weight. The greatest increases were obtained for plants continuously exposed to 125 or 250 MT. A stimulating effect on the first stages of growth of barley seeds was found for all exposure times studied. When germinating barley seeds were subjected to a MF of 125 MT for different times, 1, 10, 20, and 60 minutes, 24 hours, and chronic exposure, increases in length and weight were observed. Plants of pea exposed to 125 or 250 MT stationary MF generated by magnets under laboratory conditions for 1, 10, 20 minutes, 1 hour, 24 hours, and continuous exposure were longer and heavier than corresponding controls at each time of evaluation. The major increases occurred when seeds were continuously exposed to the MF. That's a very important statement. The major increases occurred when seeds were continuously exposed to the magnetic field. The geomagnetic field and plant evolution. Along with gravity, light, temperature, and water availability, the GMF has been present since the beginning of plant evolution. Apart from gravity, all other factors, including the GMF, changed consistently during plant evolution, thereby representing important abiotic stress factors, eventually contributing to plant diversification and speciation. Mass extinction events profoundly reshaped Earth's biota during the early and late Mesozoic and terrestrial plants were among the most severely affected groups. Several plant families were wiped out, while some new families emerged and eventually became dominant. The behavior of the GMF during the Mesozoic, and late Paleozoic, I guess, or more precisely between 86 and 276.5 million years, is of particular interest. Its virtually dipole moment seems to have been significantly reduced compared to today's values. In Earth's history, the GMF exhibited several changes of magnetic polarity, with the so-called geomagnetic reversals or excursions, characterized by persistent times with the same polarity. They occurred some 100 times since Earth's formation, and the mean time between a reversal and the next one has been estimated around 300,000 years. Because the present normal polarity started around 780,000 years ago, and a significant field decay has been occurring during the last 1,000 years, an imminent geomagnetic reversal would not be so unexpected. In the course of biological evolution, the fossil record tells us mass extinction occurred several times. One of working hypotheses to explain such mass extinctions is the case of the GMF when the geomagnetic pole was reversed. Because the strength of the GMF is strongly reduced during polarity transitions when compared to stable normal or reversal polarities, we recently proposed that these variations might be correlated to plant evolution. We do not have measurable records of GMF polarity reversals before the late Jurassic. Therefore, we can only compare variations of GMF polarity with diversion of families and orders of angiosperms in the tertiary and Cretaceous periods. Pre-sowing treatment of corn seeds with pulsed EMFs for 10, 15, 30, and 45 minutes improved germination percentage, vigor, chlorophyll content, leaf area, plant fresh and dry weight, and finally yields. Seeds that have been exposed to MF for 30 and 45 minutes have been found to perform the best results with economic impact on producers' income in context of a modern, organic, and sustainable agriculture. Various combinations of MF strength and exposure time significantly improved tomato seed performance in terms of reduction of time required for the first seeds to complete germination, time to reach 50% germination, time between 10 and 90% germination with increasing germination rate, and increased germination percentage at 4 and 7 days, seedling shoot and root length compared to the untreated control seeds. The combinations of 160 MT for 1 minute and 200 MT for 1 minute gave the best results. Higher germination, about 11%, was observed in magnetically exposed tomato var seed than in non-exposed ones, suggesting a significant effect of non-uniform MFs on seed performance with respect to RH. The effect of pre-sowing magnetic treatments was investigated on germination, growth, and yield of okra, with an average MF exposure of 99 MT for 3 and 11 minutes. A significant increase was observed in germination percentage. A number of flower per plant, leaf area, plant height at maturity, number of fruits per plant, pod mass per plant, and number of seeds per plant. However, contrasting results have been reported. For instance, the mean of germination time of rice seeds exposed to one of 2 MFs at 4 different times, 1, 10 minute, 20 minute, 1 hour, 24 hours, or chronic exposure, was significantly reduced compared to controls, indicating that this type of magnetic treatment clearly affects germination and the first stages of growth of rice plants. Possible mechanisms of magnetoreception. For a number of years, laboratory studies on the biological effects of MF have demonstrated that the fields can produce or alter a wide range of phenomena. Explaining the diversity of the reported effects is a central problem. In recent years, the following types of physical processes or models underlying hypothetically primary mechanisms of the interaction of MF responses in biological systems have been proposed. A. Classical and quantum oscillator models. B. Cyclotron resonance model. C. Interference of quantum states of bound ions and electrons. D. Coherent quantum excitations. E. Biological effects of torsion fields and accompanying MF. F. Biologically active metastable states of liquid water. G. Free radical reactions and other spin mechanisms. H. Parametric resonance model. I. Stochastic resonance as an amplifier mechanism in magnetobiology and other random processes. J. Phase transitions in biophysical systems displaying liquid crystal ordering. K. Bifurcation behavior of solutions of non-linear chemical kinetics equations. I. Radiotechnical models in which biological structure and tissues are portrayed as equivalent electric circuits. And M. Macroscopic charged vortices in cytoplasm. Furthermore, mechanisms combining these concepts and models cannot be excluded. There's some typos in this. Observation of resonance effects at specific frequencies combined with new theoretical considerations and calculations indicate that birds use a radical pair with special properties that is optimally designed as a receptor in a biological compass. This radical pair design might be realized by cryptochrome photoreceptors if paired with molecular oxygen as a reaction partner. Therefore, several considerations have suggested that cryptochromes are likely to be the primary sensory molecules of the light-dependent magnetodetection mechanism, which has been suggested to be radical pair-based. In plants, cryptochromes control different aspects of growth and development, i.e., involvement in deethylation responses such as inhibition of hypocotyl growth, anthocyanin accumulation, leaf and sotiledon expansion, transitions to flowering or regulation of blue light or regulated genes. In Arabidopsis, cryptochromes are encoded by two similar genes, blah 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 blah, like plants undergo a light dependent electron transfer reaction known as photoactivation that leads to a photoreduction of the flavin cofactor. Oh my goodness, I feel like I'm having a stroke reading these words. Particular attention has been paid to the potential role of cryptochrome as a plant magnetosensor. Experiments on Arabidopsis have suggested that magnetic intensity affects cryptochrome-dependent growth responses. But, as discussed above, these reported cryptochrome-mediated MF effects on plant growth could not be replicated in an independent study. These findings would be... Sorry, I scrolled a little bit. These findings would be very important if they turn out to exist and be independently replicable. Since even though magnetic responses do not seem biologically relevant for the plant, they would show in principle that biological tissue is sensitive to the MF responses that are linked to cryptochrome-dependent signaling pathways. They could thus confirm that the ability of cryptochrome to mediate MF response. Well, I said that, but you know what I mean. The claimed magnetosensitive responses can best be explained by the radical pair model as Arabidopsis cryptochromes form radical pairs after photoexcitation. And these experiments might reflect common physical properties of photoexcited cryptochromes in both plants and animals. I'm going to skip this one here. Recently, a combination of quantum biology and molecular dynamics stimulations on plant cryptochrome has demonstrated that after photoexcitation, a radical pair forms, becomes stabilized through proton transfer, and decays back to the protein's resting state on time scales, allowing the protein in principle to act as a radical pair-based magnetic sensor. Furthermore, the elimination of the local GMF weakens the inhibition of Arabidopsis hypocotyl growth by white light and delays flowering time, basically removing the local GMF weakens the growth and delays flowering time. The expression changes of 3 Arabidopsis cryptochrome signaling related genes suggests that the effects of a near null MF are cryptochrome related, which may be revealed by a modification of the active state of cryptochrome, and the subsequent signaling cascade plant cryptochrome has been suggested to act as a magnetoreceptor. Conclusion and perspectives. Revealing the relationships between MF and plant responses is becoming more and more important as new evidence reveals the ability of plants to perceive and respond quickly to varying MF by altering their gene expression and phenotype. The recent implications of MF reversal with plant evolution opens new horizons not only in plant science but also to the whole biosphere, from the simplest organisms to human beings. Magnetotactic bacteria are a diverse group of microorganisms with the ability to orient and migrate along GMF lines. The avian magnetic compass has been well characterized in behavioral tests. Magnetic alignment, which constitutes the simplest directional response to the GMF, has been demonstrated in diverse animals including insects, amphibians, fish, and mammals. Concerns of possible biological effects of environmental EMFs on the basis of energy required to rotate the small crystals of biogenic magnetite that have been discovered in various tissues have been discussed. The overall picture is thus a general effect of GMF on life forms. Life evolved on Earth along changes in the GMF life history. Any other environment lacking a GMF is expected to generate reactions in living organisms. These concerns becomes urgent questions in light of planned long-term flights to other planets. Understanding GMF effects on life will provide the fundamental background necessary to understand evolution of life forms in our planet and will help us to develop scientific recommendations for design of life support systems and their biotic components for future space exploration.